Hello everyone, welcome to my channel CAPSA. Today we will learn about a topic related to microbiology, that is microbial growth curve. Okay, so today's topic is microbial growth curve. Why we use microbial growth curve, or in which case microbial growth curve are very much useful. It actually used to study differential effects of media, genetics, stress on microbial population growth. So, why used? It is mainly used to study differential effects of media, like culture media, okay, genetic stress on microbial population growth etc. Now, how growth curve arised? Okay, so how growth curve arises? Actually, it has been determined that in closed system or like batch culture, bacteria will grow in predictable pattern that results in a growth curve. Closed system or batch culture means where no food, no waste, no food added or no waste material removed. So, in closed system or batch culture, bacteria will grow in predictable pattern. Resulting in growth curve. Okay. Now, what are the phases? It is consist of mainly 4 phases. Okay. Four phases. So, what are the phases? First one is lag phase. Okay. And second one is log phase, also known as exponential phase. Third one is stationary phase. And fourth one is death or decline phase. Now we will look a curve which consists of these 4 phases. So, this is x-axis, this is y-axis. In the x-axis, we will write time, and in the y-axis, we will write logarithmic or log number of bacterial cells. Now, if we draw like this type of curve, we can see a curve like this. Okay, now this phase is known as lag phase or phase 1. Okay, and this phase is known as log or exponential phase, that is phase 2, and this phase is phase 3 or stationary phase, and this phase is death or decline phase. Now we will understand this individual phase in details. Okay, so first I have told the lag phase. We will see what happened in these phases. Lag phase actually an adaptation phase where bacteria are adjusting to their new conditions or environments. Okay, so it is an adaptation phase where bacterial cells adjusting to their environment. Okay, so this is the first point. And here in this phase, they synthesize RNA, enzymes, essential metabolites, and adjusting in temperature, pH, oxygen, etc. and also repair injured cells. So, it is stated that injured cells or damaged cells have long lag period. Why long lag period? Because they must repair themselves before they can engage to reproduction. So, what we have learned in this phase, they synthesize RNA, enzymes, essential metabolites, and also adjusting in temperature, pH, etc. and also repair injured cells. And we have learned that injured cells have longer lag period. What I have said, because damaged cells need to repair themselves before they can engage in reproduction. Now, the second phase is lag phase. Second phase is log phase. That was lag and log or exponential phase. Why this is known as log phase or exponential phase? Because it shows logarithmic increase of the bacterial cell growth. Okay, that's why they are known as log phase or logarithmic phase or exponential phase. So what happens in this phase? In this phase, cells actually proceed into cell divisions, and cells in this phase are healthiest. So what happens? Cells proceed to cell division. Second, cells are healthiest. Okay. Now, for the calculation of predictability of growth, a mathematical expression is used that is known as generation time. So, for growth, generation time is used. That is stands for G. Now, what is generation time? So, generation time, actually the time takes for bacterial population to double in number. So, generation time means the time takes for the bacterial populations to double in numbers. Now, this generation time actually expressed in a mathematical formula, that is G equal to T by N. That is a very important mathematical formula. Okay, it is used for the calculation purpose. So here G is the generation time and T equal to the specific time period. Okay, and N is the number of generation. Now, to calculate the generation time, we need to get the value of N or number of generation. So, how we will get the number of generation? Another mathematical formula we should know, that is N equal to N0 into 2 to the power N, where N equal to final cell concentration and N0 equal to the initial cell concentration. Okay, these two are very much important. Those who are preparing for any kind of entrance exam in related to biology, that will be CSIR-NET or GATE in India. Okay. Now, the next phase is stationary phase. So, the next phase is the third phase, stationary phase. What happens in this phase? Bacterial populations run out of essential nutrients in this phase, okay, or the chemicals, or its growth is inhibited by its own waste products. That's why lack of physical space causing the cells, new cells, to enter into the stationary phase. So what happens in this phase? Actually, bacterial populations runs out of essential nutrients. That's why lack of physical space causing the cells to enter into the stationary phase. It will be new cells, okay. Stationary phase. And at this point, the number of new cells is being produced is equal to number of cells dying, or growth has entirely ceased. So what happens? Number of new cells being produced equals to number of dying cells. As a result, what happens? Obviously, the growth has entirely ceased, and it has been hypothesized that cells are adjust to survive for longer period of time in a oligotrophic or low environment or this kind of situation. So, this is also hypothesized that cells in this phase learn to survive for longer period of time in a oligotrophic situation. Oligotrophic situations mean low environment where nutrition will be low, the temperature, pH, everything will be low, and waste product will be more. During this phase, cells are prone to produce secondary metabolites. So in this phase, cells produce their secondary metabolites and also forms endospores. Okay, and during this phase, nucleoid condenses and the DNA becomes bound with DNA binding proteins from starved cells, that is known as DPS, to protect the DNA from damage. So in this period, in this phase, DNA protects itself from damage. Okay, now the last phase is, what was the last phase? Can you remember this? That is death phase. Death or decline. It is very much important growth curve in microbiology, is very much important topic which I always told my students to learn or to remember all the things included in the individual phases. So you need to understand clearly and you need to remember all the points. Okay. So in the death phase or decline phase, what happens? Number of viable cells decreases. So here number of viable cells decreases, and it is thought that culture conditions have deteriorated. As a result, cells harmed and unable to grow if transferred to fresh culture or fresh medium. So cells are unable to grow again if they transferred to fresh culture medium. Okay. So this is the end of my lecture. If you like my video, please do like, comment and subscribe to Grow Capsa. One like will obviously motivate me to make more videos. Thank you for your attention.